Bismillahirrahmanirrahim. We start the 9700 March 2019 paper 4-2. And hopefully we should be able to do the first 4 questions. As you all know, the paper 4 is for 2 hours, but these 2 hours are never enough, and you need to be very smart in first doing all the questions that you know and then coming back to the ones which you did not do. Because if you spend a lot of time on the question that you did not know, then you're going to waste a lot of your time. And so it's better if you first do the whole paper in an hour's time. This is sort of 2 hours, means it's 120 minutes, and you know there is 100 marks, so it is a mark per minute. And the best thing is to go to the section B, which is of course the most one, and you have to answer that. So first I advise you to read the section B. Don't if you know it very well, then of course do it immediately, because then you are sure to get your 15 out of 15 or even 14 or 12 out of 15. But if you don't know the question and you're going to struggle with it, then you need to go back to it later on. So then you should finish the section A. But if the section B is easy, then of course you do the section B first, and then you go on to section A, which is of course always has a very diversity of questions, and you have a lot of choice. Even some chapters you know very well, some you don't know very well. Now let's start with the first question, which is yes, section A. You know you have to answer all the questions. There's no choice here. Now in question 1, it says the effect of light intensity on the rate of photosynthesis can be investigated using a cut shoot of a pond plant. Now the apparatus used in the investigation shown in figure 1.1 is what? Gas collects here, then there's a test tube, then there's water containing sodium hydrogen carbonate, there's a pond plant, and there is a syringe, and there is a lamp, and there is something known as X. And as we look at the entire picture, we have a scale in centimeter, and then we have air bubble in the capillary tube, and we have the lamp here, and there's something known as X. Now X is actually a heat shield. What does it have? It has water here, and this water is going to absorb the heat coming from the lamp, so that we don't want any change in temperature of the pond weed, because we know photosynthesis is an enzyme controlled reaction, and any change in temperature would of course affect the enzyme reaction. If the temperature becomes less, slow rate of enzyme reaction. If it becomes higher, then increases rate, and then a time comes when it is denatured. So we don't want any change in temperature which could affect, because we are investigating the effect of light intensity, not the effect of temperature on the rate of photosynthesis. As we read the part 2 of the question, apparatus X shown in figure 1.1 is a thin glass container filled with water. Explain the function of apparatus X. Stopping heat from reaching the plant so temperature does not change, and temperature affects the rate of photosynthesis or the enzymes involved in photosynthesis. So as I said before also, it is actually just preventing the heat from reaching the plant so the temperature does not change. Then the part 2 of the question says, before completing the assembly of the apparatus shown in figure 1.1, sodium hydrogen carbonate is added to the water surrounding the pond plant. You see, the pond plant must have been in pond water, and the pond water might have microorganisms in it, or maybe frogs or tadpoles in it, which are actually respiring and producing carbon dioxide. But you've taken the pond plant and now you're just putting ordinary tap water in it. So you'd have to add a chemical which actually produces carbon dioxide, because you're not using pond water. And the reason why we are not using pond water is because we would not know how many microorganisms are there in it, and it could be harmful for us handling that pond water. So, and we won't know how many microorganisms are it and how much carbon dioxide does it contains. But now when we put a fixed mass of sodium hydrogen carbonate added to the water, we make a 1% solution or a 2% or a 5%. So we would know exactly how much carbon dioxide is being added or is being given to the plant, which is being released from this solution of hydrogen carbonate. So explain why sodium hydrogen carbonate is added to the water surrounding the plant in the test tube to provide the carbon dioxide. Name the gas collected, naturally would be oxygen because it's photosynthesizing. So oxygen is going to be produced and is going to collect there. Then we come to the B part of the question. The investigation was carried out with the lamp at distances of 10, 20, 30, 40, 50 from the pond plant. For each of these distances, the air bubble in the capillary tube was initially positioned at 0 centimeter on the scale, and after 5 minutes the distance moved by the air bubble was measured. The rate of movement of the air bubble was then calculated. So the air bubble was at 0 centimeter. The air bubble was at 0 centimeter and then it moved, and after 5 minutes we of course got the distance moved. So please understand this and read the question very carefully. Then it says the results are shown in figure 1.2. Now when you look at the results, what do we see? We see distance, of lamp from pond plant, and we see here the rate of movement of the air bubble. And this is in centimeter per minute, and this is in centimeter only. So please look at the units. You must always give me the units, because as we read the question further, you will understand why. Now, as we proceed in the further question, it says with reference to figure 1.2, describe. Now, I've told you this many times, describe is no biology. You have to just describe it. So you can see the description is very obvious. What is happening when we are increasing? So as light intensity increases, the rate of photosynthesis increases. And then you do a data quote. Now, what do I mean by this data quote? Now, the data quote is very obvious, as I have shown you here, is that you would give me two figures from the x-axis and the corresponding figures on the y-axis. So at a distance of 10 centimeter, the rate of movement of the air bubble is 1.75 centimeter cube per minute. I've just circled this in green, and this is exactly what I want you to quote. And I want you to give me the figures here, and you've given me here for the 10. Then another data quote, you could give me the 30, 40, 50, it's up to you, whichever you want to give. I chose 30 because it was a little simpler for me. So 30 here, and y-axis would be 1.5 centimeter per minute would be the rate of movement of the air bubble. So you gave me these two, so corresponding on the x-axis and the y-axis you read off, and that is what you got, and that is what you would have to give to me. So the data quote means you're giving me these figures, and that would get you your another mark for that. 2 says further investigation showed that at distances of less than when you sort of move the lamp, you would change the light intensity. So if you moved it even closer than 10 centimeter, the rate of movement of the air bubble was the same as in 10 centimeter. Now explain why there was no change in the rate of movement of the air bubble at distance less than 10 centimeter. Light intensity is no longer a limiting factor. So light intensity, even though you were increasing the light intensity, but that was not having any effect because it was no longer a limiting factor. Now, what other factor could be limiting? It could be temperature or carbon dioxide concentration. So any of these, if you said temperature, or if you said, so there's a slash means you wrote either of these. You could have said temperature or CO2 concentration, but slash is for us as examiners when we're checking the paper. If a student just writes temperature could be the limiting factor, well, I have to give them a mark. Or if they said carbon dioxide concentration could be the limiting factor, because there are three things which affect the rate of photosynthesis: temperature, light intensity, and CO2 concentration. So this is how we remember this, and this is how we then put it into practice when we're doing a question on photosynthesis. The C part of the question was some of the light energy absorbed by the pond plant is used in cyclic photophosphorylation, and outline the process of cyclic photophosphorylation. Now this is what I've given you in green is the non-cyclic photophosphorylation. That was not asked in the question. I'm just revising it for you. So we have PS2, PS1, and electron is excited to a higher energy level, and similarly from PS1 also it's excited, but this electron then falls here and ATP is made. And this formation of ATP is called non-cyclic photophosphorylation, while also NADPH is made. So NADPH is made also in the non-cyclic photophosphorylation. But what happens in cyclic is, I'm drawing that in red, is the electron is excited, it returns, it doesn't go ahead, it returns to PS1 and ATP is made. So this is cyclic photophosphorylation. Now inside they've asked you cyclic photophosphorylation, so I've just revised that with you. Electron excited to a higher energy level and then from PS1 and it returns to PS1, but ATP is made, and this is called cyclic photophosphorylation. Phosphorylation is when ATP is made. Phosphorylation. And there's another one we study, substrate level phosphorylation and oxidative phosphorylation. But here we're talking of cyclic photophosphorylation. Photo, why? Because light. If you look at the mark scheme, it's very obvious. Only photosystem 1, light energy absorbed by primary pigment, electrons excited to a higher energy level, emitted by the primary pigment, electron carrier goes through the electrons, goes through the electron carrier, passes along, etc. Chemiosmosis take place, and then ATP synthase is involved, and all the hydrogen ions are pumped through the ATP synthase, and electrons return to photosystem 1, or if you said P700, or if you said primary pigment. So all these is a very large marking scheme, and you only have to give me 5 points out of all these to get your 5 out of 5. I'm not saying you have to give me all these points, you only have to give me 5 points out of all these points to get your 5 out of 5. So this finishes the first question, which was a total of 13 marks. Now question 2, A part. The hormone glucagon is an example of cell signaling molecule. Table 2.1 lists the main events that occur when the blood glucose concentration decreases below the set point. The events are not listed in the correct order. Now all you have to do is read these, and when you read them, you should be thinking which one would come first. So first is adenylyl cyclase enzyme activated. Cyclic AMP activates an enzyme cascade. Glycogen stored in liver cells is broken down to glucose. Blood glucose concentration increases. Glucagon is secreted by alpha cells in the pancreas. Conformational change to glucagon receptor causes G protein activation. Active adenylyl cyclase acts on ATP to produce second messenger. Glucagon signal is amplified. Glucose diffuses out of the liver cells through GLUT transporter proteins. Glucagon binds to receptors in the cell surface membrane and cyclic AMP is formed. But you have to put these in the correct order. Now, if you did not know this, you couldn't be really guessing all this. This is not a guessing game. You had to really know this part of the syllabus. Now, the correct order was J, F, A, G, B, H, C, I. So this was the correct order. You need to go back and you need to see if you have really understood this, and if you have not, then you need to go back and revise this and then see how you're going to figure this out. Now let's look at the B part of the question. An investigation was carried out to measure the rate at which glucose is provided for respiration from 3 different sources of glucose. Number 1, amyl. Glycogenolysis. Glycogenolysis, I've told you before also, is something of the breakdown. So breakdown of glycogen will release the glucose. So the breakdown of glycogen, they've given you this even if you didn't know it. Then the third term which they've used is gluconeogenesis. Gluconeogenesis is production of glucose from non-carbohydrate molecules, like proteins or fats, so non-carbohydrate. So after a person ate a meal, now we need to concentrate the rates at which glucose was provided for respiration from the 3 different sources were measured at regular intervals over a 24-hour period. During this period, no food was eaten. So figure 2.1 shows the results of this investigation. As you can see from this graph, this is time after the meal in hours. So this is 0 is the time, and then 2 hours, and then 4 hours, and then 6 hours after the time after the meal in hours, and rate at which glucose is provided for respiration is a gram per hour. So initially you see how from the meal the glucose provided was more, and then it's decreasing, and then by this about has come to zero, just about 5, nearly less than 6 hours. Then from glycogenolysis we see that it increases and then it decreases. And then from, if you look at the third one, from gluconeogenesis, it increases by about 7 hours, and then it increases to the 24 hours. So you need to look at the graph very carefully, and then you need to explain this to yourself, how from the meal at 0 hours it was 40 something, and now it is decreasing to 0 by about 5.5 hours. And then from glycogenolysis increasing at about 3 hours, and then increases up to 8 hours, and then decreases. And gluconeogenesis starts at about 8 hours after the meal. So do this question with yourself, and then you need to answer the related questions on the graph. What is the question asking you? The question is saying state the time after the meal when the rate at which glucose was provided from the meal for respiration. Now, this is the one which is the meal for respiration, right? And then it said was the same as the rate at which glucose was provided from glycogenolysis. Now, this is glycogenolysis. Now, this was the point where it intersected, and this is what you had to read off, and that was 5. So, I'll just explain this again. State the time after the meal when the rate at which glucose was provided from the meal for respiration, from the meal, was the same as the rate at which glucose was provided from glycogenolysis. So you have to see where they both intersect, and that was at 5 hours. So please understand the question and then answer it. Then it said in the part 2, state the first time after the meal when all of the glucose for respiration was provided by gluconeogenesis. Let's look at the graph again. The first time after the meal when all of the glucose for respiration was provided by gluconeogenesis. So you see this graph, and you see it is at this point, which is 20 hours, when now meal story is over, glycogenolysis story is over, and now only this is continuing. So at this point you had to give me this point, and this is why it said at 20 hours all the glucose for respiration was provided by gluconeogenesis. Then part 3, name the homeostatic mechanism by which blood glucose concentration is maintained at a set point. That was negative feedback. That wasn't very difficult. In humans, then part 4, in humans, carbohydrates such as glucose are not the only respiratory substrate. Name 2 non-carbohydrates respiratory substrates in humans. You know it's lipids or fatty acids, or you could have said amino acids and proteins. Now, starting question 3, during an immune response, only B lymphocytes with receptors that are specific to the antigens present are activated. Activation occurs when an antigen binds to a receptor of a B lymphocyte. Now, this is all what we studied in the A1, and this is now something we are recapping. Activated B lymphocytes grow in size, then divide by mitosis. Many further mitotic cell divisions occur, increasing the number of B lymphocytes with receptors specific to the antigen. Eventually, cells produced in this process will develop into either plasma cells that secrete antibodies or memory B cells. Again, all this information we know from the AS level. Figure 3.1 is a summary of B lymphocyte activation and the events that follow. So you need to look at this very carefully. and you need to analyze it. So antigen, receptor, B lymphocyte that has not yet been activated, binding of antigen and activation, cell growth, mitosis, and then of course we see this arrow coming here, repeated cycles of cell division by mitosis. And then of course we see how the antibodies are being made, develops into number one plasma cells that produces and secretes antibodies and memory B cells. And you can see the antibodies are drawn here as well. Now this is figure 3.1. You see here the development of plasma cells and memory B cells in this process depends on transcription factors. Explain the role of transcription factors in gene expression in eukaryotic cells. First of all, transcription factors always bind to DNA, either as the promoter or the enhancer, and allows RNA polymerase to bind to DNA so that there where it's going to start transcription. Because you see, RNA polymerase is absolutely essential to bind to DNA so that then it can result in making the mRNA. So it allows RNA polymerase to bind to DNA or the gene, or you can see the promoter area, because the RNA polymerase only always recognizes the promoter area, and that's where it binds. And if we don't want to make that protein, well, we'll block that somehow so that the RNA polymerase does not bind. Then regulates, inhibits, or initiates transcription, and so gene expressed at the correct time. That is what transcription factors do, that they result in the gene expression at the correct time when the body needs it, whether it's of any organism, but when it is needed, then it should be expressed. We should not waste amino acids in making proteins which are not needed at that time. As we come to the B part of the question, transcription factors are proteins. Genes that code for proteins can become mutated. Describe how the different types of gene mutation can cause changes in the protein that is synthesized. So number one, there could be base substitution, changes in the triplet codon, base deletion or insertion, frame shift, change in the primary structure of the protein will be affected. Maybe one amino acid, maybe two, maybe three are affected. Change in the tertiary structure, this changes the protein function. Naturally, if an active site is not made of the right kind of depression, then that enzyme will not work. Maybe there's a new stop codon, and then it could result in a very short protein if it's in midway, and only if it's made up of say 100 amino acids and the stop codon comes after 30 amino acids, well then there's going to be just 30 and then nothing. So only a short protein or maybe even no protein is produced. So this is what we had to talk about, mutated, when the protein, how the code for proteins can become mutated. Then part C of the question, BLIMP1 is a transcription factor that is essential for the development of plasma cells and memory B cells in the process shown in figure 3.1. BLIMP1 reduces the synthesis of C-MyC in B lymphocytes. C-MyC is a protein that is required for the mitotic cell cycle to continue. So BLIMP1 is the transcription factor, and it is essential for what? The development of the plasma cells and the memory cells, and BLIMP1 reduces the synthesis of C-MyC. And C-MyC is a protein that is required for the mitotic cell cycle to continue. So suggest and explain how a mutation in the gene coding for BLIMP1 can prevent the development of plasma and memory B cells. So first read the question, understand the question. So if I say mutation in the gene coding for BLIMP1, so BLIMP1 is not synthesized. Expression of C-MyC continues, because BLIMP1 reduces the synthesis of C-MyC. So now that there is no BLIMP1, so C-MyC, expression of C-MyC continues. Synthesis of C-MyC is increased because BLIMP1 used to reduce the synthesis of C-MyC. This is what we're talking about. This is what we need to read in the question. BLIMP1 reduces the synthesis of C-MyC. So BLIMP1 is not synthesized. Expression of C-MyC continues. Synthesis of C-MyC increases. B lymphocytes continue to divide by mitosis, and then differentiation and specialization is prevented. Then let's come to the D part of the question. Microarray can be used to analyze the effect of transcription factors such as BLIMP1 on gene expression. Describe how a microarray is used. Now, this is a very direct question. We've done this in a previous paper as well. mRNA extracted from cells, mRNA then, of course, used to synthesize cDNA, then cDNA tagged with a fluorescent dye, hybridizes with a probe. Each probe is unique to a different gene, and fluorescence will indicate whether the gene is expressed or not. And of course, you can give me a little more about the details of the probe on if the use of the UV light to see how the different colors are expressed. So 4 marks, 4 marking scheme points, direct question, very simple. You need to learn these points and you need to do any question on microarray. Now we come to the last part of this question, and it's BCL6 is another transcription factor found in B lymphocytes. The effect of BCL6 on gene expression was compared to two samples of B lymphocytes. Sample 1 consisted of B lymphocytes that were producing BCL6, and sample 2 consisted of B lymphocytes were not producing BCL6. So again, you see in the question they give you some information. You're supposed to read that and filter some of it and then think of the possible answers. Suggest why a microarray is suitable for identifying the function of the transcription factor BCL6 in these two samples. Now, transcription factors, you tell me what are transcription factors. So transcription factors regulate the expression of the gene. Because you see, when transcription takes place, then automatically translation is going to take place. So that protein is going to be made. Otherwise, it won't be made. No transcription, no translation, no protein will be made. So microarray can detect the expression of the gene. So the first point was transcription factors regulate expression of gene. Second was microarrays can detect expression of gene. Third is difference in fluorescence between the two samples, because one you can tag red, one green, and then if you get an orange, you see both of them are being expressed. And difference in gene expression indicates regulation by BCL6. So if we know how, this one, of course, the sample B consists of that were not producing BCL6, and this was producing BCL6, sample 1. So then we can know how they regulate it, how they are going to. One was without it, so that was a very good way to compare them, because sample 1, the B lymphocytes were producing BCL6, but the sample 2, they were not producing BCL. So difference in gene expression indicates regulation by BCL6. So of course you got to know a lot more by analyzing both the samples, and that of course gives you a little more information. Now that finishes this video, because I think we can only do these questions. We've only been able to do 3 questions, and the next video will contain the rest of the questions which are left from this video. Thank you very much.